Dr. Katja Tummler from the University of Berlin. And thank you very much, Katja, for being with us. I'm really happy to have Katja to be visiting the laboratory. For a long time, our laboratory has been working on this central metabolism, and we wanted to try to make a dynamic model and to work some of other aspects. And so Jimena Martinez made the stage in Eda Klip's laboratory during 9 months last year, and they were able to make a model, and we now have another way to look at metabolism, thanks much to Katja that helped Jimena to understand the way to do the work, how was this approached. So I thank her very much for that, and she will stay in the laboratory for a few days so we can talk and work ahead in this model. And I'm really happy. It was really a very good opportunity for us. But today and next Friday, in 3 days, Katja will give the seminar at the Institute of Cell Physiology on how are they going to construct a model for a whole organism. I tell you so that you come to the Institute to hear her seminar. And today she will talk to us about the work she's doing in Eda Klip's laboratory. And I really welcome you very much, and thank you for being with us and for all the help you have given to Jimena to go ahead with this kind of research we would not be able to do without the help of a laboratory like that of Eda and with Katja's help. Thank you.
Well, thank you very much for this very flattering introduction. I'm also very happy to be here, and want to thank you again as well for inviting me. And so in contrast to the first two talks, I'm happy to give you like another aspect as well of systems biology, because the first two talks were very much like covering the ohms somehow, like the structure ohm or so, if you want to call it like that, to understand like a general set of functionalities in the cell. And what I do is more like a bottom-up approach, you could call it, where we really look in detail at a specific pathway or a specific functionality. And so that was work that I did during my PhD, and it was in mycobacteria. So I'm going to give you like a little motivation first why this is important to know. So this is how I usually start my talks when I give them in Berlin, because people are not really aware of the tuberculosis. Like the tuberculosis is a disease that is caused by Mycobacterium tuberculosis, and people are not really aware of that tuberculosis is really still a problem. Like because it's in Germany nobody has it, and it's not very known that it still exists. But if you look at the map of the world, it's very clear that it's still a big problem. And just to give you some numbers, it's like 1/3 of the overall population of the world that is somehow in one way or the other infected with Mycobacterium tuberculosis, and active tuberculosis causes more than 1.5 million deaths every year. So it's quite important to still fight this disease. And I just want to give you, for an introduction, a quick overview of how you get infected with tuberculosis. So you have this little bacteria, Mycobacterium tuberculosis, as I already said, and it enters your body through the respiratory tract. So somebody who is infected coughs, and then the bacteria come out as an aerosol, and you inhale them, and they kind of settle within your lungs. And what happens then is that your immune system, of course, recognizes them, and this is done by the primary macrophages or the dendritic cells. And they kind of, like, this is their profession, they engulf the bacteria within a structure that is called a phagosome. And what normally happens to other bacteria within this phagosome is that they are then degraded. So this is like a mechanism of host defense, that in these phagosomes the bacteria will die. But however, Mycobacterium tuberculosis has somehow found a way to avoid this killing by the macrophage. There are various mechanisms by how it does that. And because of that, they cannot be killed. There's like a very sound structure forming within your lungs that somehow contain the bacteria, but always still contain active bacteria. So you will live with the disease for a very long time, but always have bacteria inside you. And depending on the status of your immune system, there are several ways how this can go on. So you can either be like very strong, and at some point you will get rid of these bacteria, but it's like a rather long process. when you have like an intermediate strength of your immune system, this whole thing goes into a state called latency, and the bacteria go into a dormant state where they don't replicate anymore, and there they can be in your body basically for your entire life. And if at some point you should have some like immunodeficiency, as is often the case if, for example, you're co-infected with HIV, then this whole structure can break up again because this immune containment somehow gets weaker, the structure breaks up and you get contagious again. And this can be like after many, many years. So the key to basically eradicating this disease is also understanding how these cells can actually survive within this very harsh environment. And if we zoom a bit more into this macrophage, like this is my little sketch of it, and this is an actual electron micrograph of a macrophage, and you see like the phagosome here in white, and all these dark ellipses are the mycobacteria. So they are really packed within the cell. And of course, as you can imagine, this is not like a very this is a very hostile environment. So they don't have much to eat, and they are constantly fighting the immune cell somehow. So in order to understand how these bacteria can survive there, of course, you have to look at the host defense, which a lot of people are doing. Like the macrophages are actively kind of trying to defeat the cells within the phagosome by lowering the pH or like secreting proteases within this compartment. But of course, as I said before, the bacteria have found ways to circumvent that. But on the other side, and this is what I want to talk about more today, is they also have a very strict limitation in nutrients. And this is also one reason why they don't grow anymore and go into the state of dormancy, because of course in this phagosome there's not a lot of nutrients, but a lot of bacteria. So they somehow compete for the available nutrients. They are very like different kinds of nutrients, and they somehow try to scrape a living from the little amount of nutrients that are there. And the most prominent hypothesis is that they also can live from lipids that they directly get from the macrophage. So, as I already said, like in order to understand how they can survive within these harsh metabolic conditions, we really want to understand the metabolic routing, so to say, of the mycobacteria, and also what makes its metabolism so fundamentally different from other bacteria, such that the macrophage cannot clear it, and it can basically still live in the phagosome. You know, so this is like a general sketch. You see here the central carbon metabolism that I will focus on. And as I said, it can take up a lot of different nutrients, and all the red lines represent the metabolic network. And if we zoom in further, of course, each of these lines is much more complex and represents like an enzymatic reaction that, in the most easy sense, transfers the substrate into a product. Now it's interesting, of course, to understand how these fluxes can be modified, so to say. What are the most influencing factors that you would need to account for when you want to understand the metabolic flux? And we call the flux V always. And of course, there are like several things that you can think about. Many are very, very easy. Like, for example, of course, when you want to have an enzymatic reaction, you need a lot of enzyme. So the enzyme amount will somehow influence the amount of flux you have. But also, of course, how good of a catalyst is the enzyme will influence it. And then you have like more complex factors that also will shape the flux, which is, for example, saturation or thermodynamics or more complex features. And what we do, because we are a theoretical lab, we try to think of each of these things as some kind of mechanistic action and try to formalize it into some mathematical rule. And I will exemplify this here in the saturation part, which is like probably very known to all of you. So we have a mechanistic idea. We have some enzymes that bind to the substrate and produce the product. And of course, initially, the more substrate you have, the more flux you will get, because the more product you can produce. But at some point, all of these enzymes will be occupied by a substrate, and further increasing the substrate concentration will not increase your flux anymore. And we can formalize this into a mathematical equation that you probably all know, which is the very famous Michaelis-Menten kinetics, that somehow represents that mechanistic idea what we have of the saturation. That in the beginning you add substrate, your flux increases, but then at some point it saturates out because all the enzymes are busy. Genau. And this, of course, we can do not only for saturation, but for all the other aspects that we have found to be important for the shaping of flux. Genau. And those 4 are the ones that I will be mostly focusing on. And the nice thing about having all these factors now in a well-defined mathematical manner is that you can also directly include experimental data into these formulations. And I will name some, and most of them are also used in the project that I will present. So you can directly include measurements of how much enzyme you have. You can measure the kinetic parameters of enzymes, which is, has mostly been done in the past and is not done so frequently anymore, which is a pity. You can measure the reaction flux directly, which is done like by labeling substrates and so, and then recalculating the flux. It's also a very common method, but very complicated. We also include metabolite concentrations, which are hard to obtain, but very, very helpful when you want to understand this flux dependency. And of course, we also have to think about thermodynamics. So we can also include information basically about the energetics of the reaction directly into this formulation for the flux. Genau, so now coming back to the mycobacteria. So the question I had was, I wanted to understand how this flexibility in the mycobacteria, that it can deal with very different substrates, is facilitated in detail or mechanistically. And as I already mentioned, there's like a very popular hypothesis that inside the cell, the mycobacteria also feed on fatty acids that are derived from the host. So it basically eats up the host membranes or lipid stores that are stored within the macrophages. And if you look again at our pathway map, so this is the central carbon metabolism to anybody who's not familiar with this kind of representation. So you have glycolysis here, which is basically the main pathway of sugar catabolism. You have the pentose phosphate pathway to produce nucleotides, the Krebs cycle that gives you energy, and then mycobacteria have these 2 additional pathways that are able to process parts of the fatty acid, so to say, or the end products of fatty acid decomposition, which is the methyl-malonyl-CoA pathway and the methyl citrate pathway. And we can zoom in here a bit more, and we see that they are actually very similar. So they are kind of 2 parallel pathways that consume this compound here, propionate-CoA, which is the end product of many degradation processes that take up fatty acids. And then they do like different reaction steps, different mechanisms, and the end product is succinate that can be directly fed back into the central carbon metabolism in the TCA cycle. And the question I was asking now was, why do they need these 2 pathways? Is there anything specific about them that somehow helps the bacteria to cope with this very untypical carbon source? Genau. And another thing is that this pathway is not present in human cells, so it's a very interesting target also for developing new medication. So when you go in the literature now, you find many, many studies about this pathway, and it's very preferred when actually these cells grow on carbon sources that contain fatty acids. So whenever you give the bacteria fatty acids, they will upregulate this pathway and try to catabolize all the fatty acids via this pathway. And interestingly, it's also upregulated in many infectious scenarios. So when you have a mouse model and you will look into the lungs of the mouses and extract the bacteria, they will also have this pathway upregulated. What is interesting about that is that actually one of the intermediates of this pathway is very toxic to the cell. So here you have to tightly control the concentration levels of this compound, which is called methyl citrate. And recently there was also a study that explained where this toxicity comes from. And it's very hard to see here, but it's inhibiting the pyruvate dehydrogenase in the central carbon metabolism. So it basically stops you from feeding into the TCA cycle and stops you from gaining energy. Genau, so this is like a very critical compound to be dealing with, but the cells somehow still prefer this pathway. And this is like the catabolic direction when you want to feed on fatty acids, but of course there's also biosynthetic function of these pathways, because intermediates are required to build the cellular fatty acids, so of the mycobacteria themselves. And this is also very important because they have very complex lipids, and they are also very important for the virulence, so to say. They build up like a very thick layer of lipids around them, basically, which also contributes to them being very hard to kill. So also the biosynthetic direction, of course, is important, but here it's not really known which pathway is used. So the question is now, why do you need these two pathways? And which of them has which features, so to say. So the first thing we did, of course, we tried to acquire data from for that pathways. And this was done in collaboration with the group of Uwe Sauer and Rudi Ebersold at the ETH in Zurich. And we basically designed experiments that somehow should mimic this situation. So having one nutrient in the media that somehow mimics the situation when you grow on fatty acids, which is just feeding this propionate into the media that is directly transferred to this first metabolite of the two pathways. And then we also have a condition where we feed glucose, where basically biosynthesis is required through those two pathways. I'm not going to talk about the glutamate today. So, and then we basically measured all metabolite concentrations in these two conditions, and also had a quantification of the proteins of these two pathways. So what we can now do with this information, or what the first hypothesis was, is acceptar. Okay, it wasn't good to accept it. Yeah, it increased to such high levels. Ah, no, what happened? Okay. Okay, so I'm very sorry for the interruption. Good, let's go back. So we have this set of data for these two different conditions, catabolic and anabolic, basically. And the first thing we can analyze is the thermodynamics, for which you, in the beginning, only need the concentration of the metabolite. So I'll also give you a short introduction into reaction thermodynamics, which are basically governed by this quantity here, which is the Gibbs free energy of reactions. And you have some standard term that characterizes the type of reaction you have, and then of course it's influenced by the concentrations of your metabolite. So if you have a lot of substrate, your reaction is more likely to run into the forward direction. And this is encoded here in this quotient Q, which is basically the ratio of substrate and product concentrations. And the thing about this this Gibbs free energy is that when it equals 0, your reaction will be at equilibrium. And that doesn't mean that there's no flux, but that the forward and the backward flux will basically compensate each other such that you don't have a net production of your product. If the delta G is negative, that means that your reaction can run in the way that you want it to run, in the forwards direction, and that your flux will be larger than 0. As soon as this term becomes positive, your reaction can only run into the reverse direction, so you cannot produce the product anymore that you wanted. And of course, the more negative this term comes, the easier it is for the cell to let it run in that direction, because the more energy is released on the way. And this is relatively straightforward when you only look at one reaction. But if you look at a pathway, there's another aspect that you need to facilitate the flux through the whole pathway. So you cannot regulate the substrate and product levels individually of each reaction, but you have to guarantee that the flux through the whole pathway is possible. And then, of course, it doesn't make much sense to make one reaction very negative, so to make the gradient very steep, because then all the other reactions don't have a lot of energy to dissipate anymore, and you will have a very shallow thermodynamic gradient, and all the pathway can work only very slowly. So this is some kind of optimality problem, that you want to distribute all the energy loss, so to say, equally. Genau. And when we look at our two pathways, we actually see that there is no significant difference in the direction that consumes the fatty acids. So I always put here, like this is basically the same diagram as here for each of the pathways, and here is the delta G of the shallowest reaction, so to say. And you see that they don't really differ a lot. So that wouldn't be for the cell a reason to prefer one pathway over the other. However, if we look into the biosynthetic direction, we see that actually only for one of the pathways the delta G becomes negative and the other one cannot run in the backward direction. So the term here becomes positive. So that means that the cell definitely will need this pathway for the biosynthetic direction in order to be able to produce all its lipids that it needs to survive. Okay, and another conclusion is here that thermodynamics are not alone able to explain why the cell has these two pathways. So we can go further and say, okay, we have thermodynamics, but we can also look at the enzymes themselves, because of course that is also an aspect of shaping the flux, so to say. And this also relates back to the thermodynamics, because as I told you before, when you are very close to the equilibrium, so if this gradient here is very close to zero, you will have a lot of backward flux that doesn't go into the direction that you want to go, basically. So you need a really, really large amount of enzyme in order to be able to facilitate some flux into the right direction. And this, of course, is very, very expensive for the cell. You need to produce a lot of enzyme in order to get really little flux. And additionally, not all enzymes, of course, are the same size. So if by chance, like this enzyme would be a very, very big one, it would not be very good for the cell. Like, it would need to invest a lot of its resources in order to sustain the flux to this pathway, and that would maybe evolutionary not be so efficient. So the thing we could ask now first is any one of these pathways cheaper? So should the cell prefer any one of these pathways? And maybe are there also cooperative effects that if you take a bit less protein in that one pathway, you will somehow promote the other pathway? And the analysis we did here is basically take a fixed amount of protein mass, which I just took as being 1% of the overall protein of MTB, and then run an optimization algorithm that distributes this protein mass between the pathways and see where to invest it best, so to say. So on this axis is where the protein goes, so to say. So if it's 1, it will be fully in the yellow pathway, in the methyl citrate pathway, and if it's 0, it will be fully in the methyl malonyl-CoA pathway. And the lines indicate the flux, basically, so the higher you get here, the more flux you will have. And you see that there's, first of all, no cooperative effect, like these lines are lines, basically, not curves. And you see that under the biosynthetic glucose conditions, the methylmalonyl-CoA pathway can produce a bit more flux. But if you look at the catabolic conditions, it actually turns around, so that suddenly when you put all the protein in the other pathway, you will be able to get a higher flux. So by including basically this information about the sizes of the proteins, and in addition to the thermodynamics, we can say, okay, here we already see that it's somehow condition dependent, right? So under one condition you would want to prefer this one pathway, and the other condition you would want to prefer the other one. But still, it doesn't really give you an idea about, or like an answer to the question why to keep both, because you could say like the cell somehow can adapt to one condition or the other and just get rid of the other one. So the next step would be, okay, so maybe it's really in the dynamics, in the saturation term that is important. So we again harnessed or asked our experimental partners to perform some more experiments, and this time in a dynamic fashion. So we didn't only look at the steady state concentrations, but we basically took the cells out of the medium that they know and subjected them to the opposite one. So for example, we took them out of glucose and put them into propionate medium, so such that they really, in a very quick manner, need to reroute all the flux. And then we measured like within 160 seconds how the metabolite concentrations change over time. And this is also like a short enough period that we can assume that enzyme concentrations stay constant. Genau. And then we actually see that the dynamics make the difference. And so this will always be, the dots are the experimental measurements, and the lines in the shaded area are a dynamic model that we did of the two pathways. So the lines are our predictions. And here again you see the initial component of the two pathways, and here the metabolites of the methylmalonyl pathway, and here of the methylcitrate pathway. And the first thing you notice is that the metabolite levels are really, really upregulated when you grow them on the catabolic carbon source in only one of the pathways. And then when we look at the dynamic condition, where we switch them from one carbon source to the other, we see that actually only one pathway reacts, and it's not the one that was upregulated. So we see that we get a very dynamic response. So the propionate-CoA levels drop when you take the propionate out, and they increase when you add the propionate suddenly. And we see that this response is only transmitted to one of the pathways. And this is actually not the one that is preferred in vivo, which is really surprising. And due to the fact that we now have this model that somehow captures this behavior, we can ask the model, like, can you explain why this happens and why this is important? And here I show you again the equation. So all these lines are produced by setting up, for each of the reactions, setting up this kind of equation and estimating all the unknown parameters. And then, of course, we can again do this kind of Michaelis-Menten plot that I showed you before, which basically shows you how the flux of the first enzyme, I'm only showing the first enzyme here, depends on the concentration of substrate. And this now includes all the aspects that I talked about before, the saturation, the thermodynamics, and also the enzyme amount that was measured. And then you see that this one pathway that can react dynamically basically to changes in the nutrient supply can very flexibly adapt its flux depending on the concentration of propionate-CoA. So when the concentration increases, like this dashed line here is the actual concentration that was measured within the cells. And when you increase it a little bit, like the flux will go with it, and when you decrease it, the flux can actually invert its direction. So it can become negative, and then the biosynthetic capacity, so to say, of this pathway can be switched on. And the other pathway doesn't react much, actually, to the concentration. And if you zoom in further in which of these parts of the flux factors, so to say, is responsible for this behavior, you see that actually it's the thermodynamic term, which also makes sense because this is the only one that can become negative when the concentrations change. So you see that the saturation term is almost at 1. That's also the highest value it can reach, so the pathway is very saturated. And then all the switching behavior only depends on the thermodynamic term. So you can say that it's like really properly thermodynamically regulated. Genau. And when we look at the other condition, actually this dependency on the thermodynamics becomes very negative for the pathway. So now the propionate-CoA concentrations are higher, and then this is the situation where you really want to take up the propionate-CoA. So you want to live on it and somehow get your carbon from it. So it's actually really, really bad if your flux runs into the other direction, in the biosynthetic one, because then you cannot use the propionate-CoA anymore. And because of this dependence on the thermodynamics, this is actually what happens in the orange pathway, in the methylmalonyl-CoA pathway. So this is not very good for the cell to have, so to say. If it would only have this pathway, in some situations where you would have some fluctuations around this value, the flux could actually invert and would be very bad for the cell. And for the other pathway, for the professional pathway that we find to be upregulated in many experimental conditions, you see that now under this experimental conditions that somehow mimic the reality, suddenly the flux depends on the propionate-CoA concentration, and then it gives you a way to like reliably take up this metabolite under this condition. Genau. And I didn't put that here. And this change between this and that situation is mostly facilitated by actually very, very strongly upregulating all the proteins of this pathway. So the cell takes all the hassle, so to say, to produce all these proteins from anew when it knows now I really want to grow in propionate. It increases all the enzymes of this pathway, and then the flux can increase, so to say. And now we can actually harness some very old method, metabolic control analysis, to understand why this is basically the only thing that a cell can do. So I don't know if everybody knows it, but I'm still anyways going to give like a short introduction to metabolic control analysis. It's basically concerned with steady state flux distribution. So it gives you a way to learn in a steady state how the different parts of your metabolic pathway influence each other, so to say. And one of them is the flux control coefficient, which basically gives you an information about how changing an individual flux would change the whole flux through the pathway. And if you now recall how our equation for the flux looked, this can be in practice facilitated by like raising the enzyme or increasing the K-cut or so, anything that is linearly related to the flux. So this is the flux control coefficient, and then we also have the concentration control coefficient that basically says the same thing about the concentrations. When I change one flux, like for example, when I increase V2 here, how would I change all the concentrations in the new steady state that I would arrive at? And I want to show you here the flux control coefficient because it's the most easy thing to understand. So we have here all the reactions of the pathway. Here is the non-professional pathway, and here's the one that is always upregulated within the cells. And then you see that actually, so the flux control coefficient is now encoded in the color, like the more intense the color is, the higher is the control coefficient. And then you see that actually only the first enzyme of this methyl citrate pathway has control over the flux through both pathways. So it's basically the only way of the cell to somehow, under these conditions, to change the flux through these pathways by upregulating these enzymes and thereby taking all the other fluxes with it, so to say. So the new steady state that will come after I increase this enzyme here, it will be with a higher flux through the whole pathways. And that's basically it. So we, with our dynamic model and all these nice data, we were able to really in detail explain how these two pathways interact and how the cell needs them both basically to deal with this metabolic challenge of having to grow on propionate or on fatty acids derived from the cell. And in summary, we could basically characterize this pathway that is always upregulated in the infectious scenario as like, first of all, irreversible, a high capacity catabolism with favorable thermodynamics that works somehow with this on-off regulation. So the cell cranks up the production of the enzymes once it has to. And on the other hand, we have this methylmalonyl CoA pathway that can basically very, very quickly react to when the conditions are changing. So you have this as some kind of intermediate pathway that you need when something changes suddenly. And as I showed you, this is mostly regulated by thermodynamics. We also need it for biosynthesis, and we don't have this regulation of the enzymes. So I hope I was able to convince you that also like looking at the details of one specific pathway can be very, very helpful. And you can really learn a lot about that by this bottom-up approach. So thank you for your attention, and here are all the people that contributed. The Edda Clip is the leader of my group, and Clemens and Max helped me a lot with the implementation. And here's all the group that was so also flatteringly introduced in the beginning. The experiments were done by these two groups at the ETH, and with those people I collaborated a lot about the thermodynamic modeling. Yeah. Thank you very much. Sorry for the interruption in between.
Questions?
Hi, thank you for a nice talk. What would your model predict about the behavior of mutants in this in some of these pathways, and I wonder if you have tried this experimentally or modeling it in some other way.
So basically, all the knowledge that is known about these two pathways was done by knockout experiments. And let me go back to this. So what people mostly did was knocking out the genes of this more interesting pathway here, this methylcitrate pathway, because they saw that it's very, very relevant to virulence, and basically all imaginable experiments are already done in this pathway. So they knockouts, knockdowns, and then they measured basically how the concentrations change and also how the virulence changes. And all of these reactions are very, very important, so to say. So if you, for example, like knock out those ones, this metabolite will accumulate, and then the cells will die. And this is something, like I didn't show it here, but this is something that would of course also come out in the model, but it's a very straightforward prediction as well. And the other pathway is not so well studied.
Okay, and do you, but how do the mutants in the other pathway behave? I mean, is there some evidence of non-redundancy there in terms of the?
Yeah, I mean, it's basically, I've showed you that it's non-redundant basically, no? So we, you see that this pathway can also somehow compensate for the other one when you knock it out, but only to a certain level, because you see that, as I showed you with the thermodynamics, it somehow saturates at some point, so there's only so much it can do, and then at some point it will just be saturated, so to say. And this is also something people saw in experiments, that when you knock out this pathway and somehow increase the activity of this one, you can compensate for it for a while, but not forever, so to say. When the concentrations of the substrate go too high, then the cells will still die. Genau.
Another question?
This one, the yellow one, is not present in human, the methylcitrate pathway. So also the methylcitrate is actually a metabolite that you don't have in mammalian cells, I think. But this other one you also have because it's for the synthesis of lipids. It's required.
Okay, thank you.
Oh.
Do you know if someone is doing some research to inhibit some of the enzymes of the orange pathway?
To inhibit it, yeah. Some research is done on this enzyme here, which is like the isomerase, I think, because it's strictly vitamin B12 dependent, so it needs as a cofactor the vitamin B12, and this would be like a good target point, so to say, to fiddle with the activity. But on the other hand, it's not so capable, so to say, because it's not the main pathway that is used in the cells, but it's only in these cases where you suddenly change from one nutrient source to the other, which might not be even very often relevant in an infectious scenario. So the focus is really more on this pathway than on the other one. Genau, but actually if you really want to be effective, you would have to kill both of them.
And do you know anything about the evolutionary origin of these two pathways? They share a common thing, or they appear independently?
No, like this pathway occurs in almost all species, like because it's required for lipid synthesis. So you will find it in many, many species, and there's also like a lot of studies about how it evolved that I'm not like super familiar with. And this one you only find in bacteria, actually.
Okay.
So,
that's why it's always the best choice to...
Yeah, genau. And actually mechanistically they function a bit different as well. Like it looks here now, they are very, very parallel, so to say, but the functions are very different. Like one of them needs ATP to be activated, and the other one takes another carbon compound from the central carbon metabolism. So the mechanisms are a bit different.
Okay, thank you.
A nice talk, Katja.
Okay.
I have two questions.
Yeah.
The first one is the problem that always the people that work in systems biology, that sometimes the network is a sub-network of a bigger network. So my first question is how another components, another reactions, for instance, one of these metabolites can produce from another pathways. How did you deal with this problem? That's the first one. And the second one is, in terms of the metabolome, sometimes, according to technology, it's not possible to obtain all the metabolites in the pathways that you are suggesting. For this lack of information, how do you resolve the problem?
So let me first talk about the first one. I even have like an additional slide for it, I think. So the good thing is that we now live in the age of genome scale metabolic reconstruction. So we actually, these are basically large networks that people reconstructed that contain all the information about the metabolism of a certain species. So we can actually take a lot of knowledge from there. And what we do is we basically cut out our little pathway from this large network and then analyze from like the distribution of the large network what kind of leak fluxes there would be from these two pathways. And we include those, at least in a steady state manner, in our simulations, which is here these two bioreactions that I talked about. So we kind of include the need that the network tells us that you need, like for biosynthesis or for keeping up your energy storage, as boundary conditions to the model. And for the other part is, so if the metabolite concentrations cannot be measured, we cannot include them. And what we usually do is like we take some generic bounds, like we know for some concentration ranges to be reasonable within the cell, because otherwise there would also be toxic effects or so. So if that can be at some parts the only information you can include, but what we also often get is like that you can only measure the sum of some metabolites, that you can, for example, not distinguish between isoforms or so. Like for example, here for the methyl citrate and the methyl isocitrate, they just will show up the same in the mass spec. So we cannot distinguish them, and then we can include them via observables. Basically, it's like a modeling technique that you say, okay, I can only measure this pool of metabolites, and you basically make your predictions for this pool, being aware of that it's actually a sum of two of your model species, and that somehow, like it's more accurate, but it decreases your predictive value, so to say, because you cannot resolve these two species individually, but you can only say something about the sum.
I was thinking when I was listening to you about the thermodynamics laws that you were announcing, so about the thermodynamics of your system. So the general idea in thermodynamics is that the entropy should increase in your system.
Yeah.
So the point is, in your equations, how do you deal with that situation?
Naja, it's like the general laws that the entropy in the universe should increase somehow, no? But
no, but also the individual systems. So it depends on how do you define your universe.
That's like a very fundamental property of when you look at reaction or like an energy thermodynamics, that actually within the cell the entropy doesn't increase, because it takes up, I don't know, dissipates heat and so, and it, when you look at it properly, it is not the case that within the cell the entropy increases, and also not that there's some kind of limit on entropy production.
You know, that's a subject that I recently read a couple of, the review and briefly the original two papers that were published last year in two independent journals. One of them was Nature, and the idea is precisely that for a long time it's been debated, the idea that entropy should increase, but some people will say, well, this is only the universe, but locally we can keep it.
Yeah.
But so these two theoretical groups propose a new way to analyze thermodynamics, and they recognize that in a system, in an isolated system, entropy will increase if you consider that the particles of the system will share not only one form of energy, but multiple forms of energy. And every time that two particles interact, they will exchange different types of information. That's a more general subject, but so in your system, you are not considering that the entropy will increase?
No.
Okay.
No, so we
That's
go
an
like with a
interesting
very classical notation, so to say that it doesn't increase.
Okay.
Thank you.